Welcome, Dr. Witten. Thank you so much for joining us tonight. We so appreciate your time.
Thank you.
Sure. I have loads of questions for you, Dr. Witten, in our short period of time. The first one is about CBER and CDER, the Center for Drugs and Center for Biologics. You know, our community is very familiar with CDER, the Center for Drugs, and now very hopeful for gene therapy, which sort of lands in your lap. Is there a crosstalk? How do you communicate and get all of the learnings that have occurred in the antisense oligonucleotides and the steroid approvals and take that into account as you look at gene therapies?
Thanks for that question. We're very excited about gene therapy in this area. I'll just say that. And I think our communication with CDER, who has more, more experience with approvals, is important. But we've had regular communication for years. So I'll describe some of the forms of communication. They are both programmatic and also in the setting of specific applications. So we have a monthly meeting that actually includes all three product centers, CDER, CDRH, and CBER, to discuss specific neurology applications and topics of shared interest. So those could include things like clinical issues, endpoints, trial designs, and also the rationale between specific regulatory decisions. So that is an important forum for communication, so we have regularly know what is up in each other's area and can have some discussion about it. And also consultation is obviously important. So when we review specific applications, we frequently consult other FDA centers. In this case, it would be the neurology group at CDER, and their perspective, expertise, and experience are often useful in our consideration of clinical issues like the interpretability and meaningfulness of proposed endpoints. And then there's some other efforts in terms of, development of tools for product use that I think are still in development. We don't really know, how they'll be used, but we certainly are interested in the, the CPATH efforts and the area of drug development qualification program. So we do learn about those and follow along and hope to benefit as those, tools evolve.
Yes, thank you. We had a call with Krista Vanderborn from Imaging DMD, and she's talking about the qualification process for fat fraction. So we're very interested in those tools. So Duchenne is a slowly progressing disease, as you know, and sometimes it's really hard, especially in short periods of time, to evaluate safety and efficacy. So I would love for you to comment on this accelerated approval pathway that we experienced in the Center for Drugs, and how you're thinking about that in gene therapies.
Yeah, so just, to take a step back and talk about just the overall context of accelerated approval. And I agree it's an important topic. Home runs are easy to assess. Of course, that's what we always want, would want, hope for. And if you have one in Duchenne's, for example, maybe you'd see the disease stop, the strength return to normal, a normal or a near normal biopsy. And that would take only maybe a clinical trial of a year or two to be confident. But home runs are easy to assess. But the challenge is that most products are not home runs, and the benefits may be more modest. The interpretability of the study then becomes more subjective and variable. So in these situations, just as a general matter, it becomes critical to have concurrent, controlled, appropriately blinded clinical trials, and longer duration studies become even more important to be confident that the effect is real. And I just caught the tail end of the previous discussion, so I know there was pretty active, comments on some of these points. But we support CBER as a center, certainly supports accelerated approval, and the office that regulates gene therapy for Duchenne's in particular used it recently in the approval of a CAR T-cell product in oncology. So it is part of what we do. Accelerated approval relies on a surrogate endpoint that's reasonably likely to predict a clinical benefit. And our ability to rely on such a surrogate depends on having a good understanding of the pathophysiology and natural history of the disease and the role of the proposed surrogate in that pathophysiology. So for many rare diseases, those may not be well understood. Fortunately, in Duchenne's, the pathophysiology is generally well understood. The role of dystrophin in that pathophysiology is well understood. In gene therapy applications, it should be noted that the sponsors have proposed to use many versions of dystrophin as a surrogate, and the role of mini-dystrophin in the pathophysiology of DMD is less clear than the role of dystrophin. I'm just mentioning that is one of the things that we would keep in mind. So in general, though, just getting back to the general topic, if a drug company proposes to use a specific endpoint as a surrogate for clinical benefit, we would ask them to submit data to support that the proposed surrogate is reasonably likely to predict a clinical benefit. So we're always open to this option, but we need to see data to support the proposal. So I guess
Sure.
that's right.
It's hard, right, because of the slow progression, and I do understand. And so as you look at, you're doing many, or you're overseeing many gene therapy, pediatric gene therapy studies. What are the ways you're thinking about to accelerate approvals or to accelerate this process? As you know, so many people are out there waiting and hoping for certainly a gene therapy that is both safe and effective. So how are you thinking about that?
Well, that is a really good question. And let me say that one of the things that we care about and always are thinking about are any efforts to develop cross-cutting tools. And something that might be useful in more than one setting, that is not in a specific sponsor's own development program, but across the board. And so one area, as you know, that is definitely of concern to FDA and the community and the patient community also, and again, I heard a couple of questions about this in the tail end of the last discussion, is the question about neutralizing antibodies and neutralizing antibody testing. And so that is, it's important, and it's important to understand what the results mean. And it's important both for the individual product, but it's important across the field to really, understand it so you can interpret things from one setting to another and really understand, the data more generally than just what the results meant for that setting. So we have, for that kind of thing, and for a number of other topics like that, a couple of years ago, FDA stood up a group, an organization. It's a public-private partnership called the Standards Coordinating Body. Mm-hmm. And that organization looks at opportunities for standards development in the field. This is one of the topics that they looked at. That group doesn't develop standards, but they support standards development by facilitating participation of U.S. experts in standards development efforts. So I'm hoping that this topic of neutralizing antibody it is proposed as one of the topics at an international standards organization to take up. And what that means is they would develop test methods that people could more uniformly apply for that kind of testing, so you could interpret it better across the field. And there's a lot of examples like that. So the standards world is one area where I think further work in standards can enhance the field. Then there's other questions like, how do you leverage information from one development program to another? And that could be the data about the vectors. It could be information about preclinical testing. I mean, it's a general question. So one of the things that CBER is doing is partnering with the Foundation for NIH, NCAT, and NCATS on the so-called Bespoke Gene Therapy Consortium, BGTC. And the objective of that group is to look in a consortium framework at a common set of procedures in a pre-competitive environment to see whether that can drive innovation and promote access to individualized gene therapies. And it's hoped that we will learn something from that effort that will, the phrase "a rising tide lifts all boats," that in that effort to figure out how to develop, test, and deliver therapies to very small diseases, we'll learn things that'll help with larger development programs in the community as a whole. So it can be questions like, how do you leverage preclinical and CMC data, or learn about innovative trial designs that might facilitate development, and even simple things like the use of master files, procedurally, like what's the most efficient way to help manufacturers submit, those who have a platform technology. So those would be some examples of what we're doing. Or maybe that was too long-winded, maybe.
No, it was wonderful, and we can't thank you enough. I think the neutralizing antibodies, to have for patients and families to understand what the testing means and whether it applies to all serotypes, would be impressive. I think how to handle the complement response when these children get a virus and have a response to that, and if that could be generalized, and certainly data and looking across. I think all of these things are so important for our families and for the world of rare diseases. I can't thank you enough, Dr. Witten, for what you do, for how you communicate with all of us, and how you're working so hard. If there's anything we can do to help you accelerate, our fingers are crossed, and we're hopeful to get a home run, or even a near home run in gene therapy. So thank you for joining us tonight, and we hope to see you very soon.
I appreciate that, and I just want to add one last word, which is we appreciate the efforts of the patient communities, and we're looking forward to seeing the guidance document as you develop it.
Thank you. Thank you. We're thrilled to be developing that guidance. Thank you again. We'll see you soon.